I'm going to be talking about graph-theoretic models in protein structure. And so first there'll be an overview of all the different kinds of graph theory and a bit of a literature review with respect to the graph theory that we've seen, networks and more literature review, and then we'll have a primer on proteins so that we're all on the same page. And then the dilemma that we face and a possible solution. That's going to overview the talk. So these two graphs, you're probably familiar with Peterson's graph here on the left. And the vertices are labeled 1 through 10. And notice that each vertex has degree 3, meaning it's adjacent to exactly 3 vertices. And there are 1, 2, 3, 4, 5 edges on the outside, 5 edges on the inside, 5 edges connecting, so it has 15 edges, 10 vertices and 15 edges. On the right, it's the same graph. It's just been drawn differently. So we still have Peterson's graph on the right, and the reason it's the same is if you look at each vertex, say you look at the vertex labeled 2, it's adjacent to 1, 7, and 3. And if you look over on the right, thank you. If you look on the right, what did I press? Whoops. Yeah, now I got to go back. That's too far. There we go. It's too complicated here. So if you look on the right, the vertex labeled 2 is again adjacent to 1, 3, and 7. So the claim is if you check every single vertex, you have not changed the adjacencies. So if you have a mapping from one vertex set to another that preserves adjacencies, then that's an isomorphism. And these are just two copies or two isomorphic copies of the same graph. Okay, so what do we do with these graphs? We typically define a lot of numerical quantities that say something about the structure of the graph. They're called graphical invariants, specifically because they don't change under isomorphism. The most obvious of these we've already done. Count the number of vertices, count the number of edges. Now the degree is a measure of a single vertex, but you could list the degree sequence, and that would be for the whole graph. Other kind of graphical invariants, like the diameter, which is the distance between two vertices is the shortest path. And so there may be multiple paths, but the shortest one is the distance. So over all pairs of vertices, you take the maximum of the distances, and that's the diameter of the graph. So if we look at these two, they have the same number of vertices. They have 17 vertices. They both have 16 edges. They even have the same degree sequence. So the 3 graphical invariants that we've mentioned so far, these are identical. But they're not isomorphic. So that means there's at least one graphical invariant by which they differ. And if you look at the top one, the distance from this vertex to this one is 6, and that's the maximum distance over all pairs, so it has diameter 6. But if you look at this one, there are no pairs of vertices that are 6. Everything's within 5. So these cannot be isomorphic. These are two distinct graphs. Okay, so let's look at some, say, interesting graphical invariants. The first one here is the independence number. So two vertices are independent if there's no connecting edge. So this set, go back. This set containing 1, 6, and 4 is an independent set because there's no connecting edge between any pair. Furthermore, it's maximal because if you try to put any other vertex into the set, it will create an edge. All the remaining vertices are connected to either 1, 6, or 4. So that's as big as you can get. So the definition, oops, I'm on the wrong, on the independence number. Hold on. Maybe I should put my glasses on so I can see my arrows. I'm sorry about that. So which way? This is back. There. That's where we are. Okay. So this 1, 6, and 4 for the independence number is the maximum cardinality among all independent sets. And so if you say that this is, therefore the independence number is 3, you would be wrong. Even though you can't increase the size of this set, perhaps you could have made a wiser choice. So instead of 1, 6, and 4, if you had chosen 1, 6, and 8 first, then you could add the vertex 3 to the set, and now you have an independent set of size 4, contradicting the first claim that the independence number was 3. So this really points out to why these values are intractable, because you can't write a computer algorithm and say, well, pick this vertex and take this one if it's adjacent to it, put it, don't use it, take this one if it's not, put it in, because the algorithm would have terminated before and returned a number of 3, which was incorrect. So the only way you can find the independence number with a computer algorithm is to check all possible cases, and which becomes intractable very, very soon. Okay. Let me sure I push the right button now. Here's another one. The domination number. Okay, so we go back. We've got 1, 6, and 4. And the domination number, whereas the independence number, we were looking for the maximum. Now we're looking for a minimum such that every vertex that is not in the set is adjacent to at least one in the set. So this is a dominating set. And so over all possible dominating sets, you want the one with minimum cardinality. So you start to see this trend, everything's maximum or minimum, or the minimums of the maximums, and so forth. Okay, so this is typically, we call it graph theory, but it's actually, we should say more specifically, it's combinatorial graph theory. Because, whoops, activity alert. We may need some of these definitions for the activity. Now, what do we do with these combinatorial measures? We usually don't find them, because like I say, they're intractable. We're not usually saying, what is this number, what is that number? Typically in combinatorial graph theory, it is mathematics, so you say, suppose we have a spherical graph. You have a graph, it has property P. You want to show that that's sufficient to say it has property Q. So something as simple as how many vertices it has n, whatever n is, and corresponding, this should be edges. So it has n vertices and n squared over 4 edges. With respect to vertices, if you know it just has those two things, then you know it has a triangle as a subgraph. That's sufficient to show that. Different kind of graph theory. So let's look at these two again. We saw this as an example of two graphs that can have the same, similar, well, actually they had the same number of vertices, same number of edges, same degree sequence, but they were different. These actually are graph representations of a molecule that satisfies this chemical equation. For every m carbon, you have 2m plus 2 hydrogens. And so the ones here of degree 4, those are the 5 carbons. And then you have the 12 hydrogens here, and then you have your 5 carbons and your 12 hydrogens here. So these represent, since we know they're not isomorphic, we know we have two distinct molecules. And that actually was the topic of the very first article that was published in the American Journal of Mathematics in 1878. It It was one really long article in which John Sylvester, the author of the article, enumerated all the possible saturated hydrocarbons by determining all the possible configurations that satisfy the chemical equations, and he enumerated them, and said that he actually claimed that there were 2 more saturated hydrocarbons than what were known at the time. And then a couple of years later, they discovered the additional 2 saturated hydrocarbons. Okay. So in chemical graph theory, which is different from combinatorial graph theory, they call graphical invariants topological indices. And the topological indices that are very well known, for example, the Balaban index or the Wiener index or the Platt index and so forth. Here's an example. The Balaban index is given by this formula. So no max and min anymore. This is a straightforward formula. And where Q is the number of edges, and you sum over edges, and the S sub I is the sum of the distances from vertex I. So it's easier if we see an example. So if we take this graph, the first thing that we do is construct this distance matrix. So in the ith row and jth column is the distance from vertex I to vertex j. So of course we have zeros on the diagonal. Distance from itself is 0. And like if you look at the first one, it's distance 1, 2, 3. Then because of the fork, you have 2 that are distance 4 and 2 that are distance 5. So the formula says out front you have this Q, which is the number of edges, which has 7. This is the cycle number, and since there are no cycles, we just have 7 over 1, or 7. You're summing over edges, so there should be 7 terms, 1 for each edge. And here, if you look at the edge 6, 8, that means you take the sum of row 6 times the sum of row 8 for this product in here. And that gives us the Balaban index for this graph is approximately 3.1, a little more than 3. So topological indices are used extensively. We don't say chemical graph theory as much now. We say computational chemistry. And computational chemists use these QSAR models, where QSAR is what quantified structure-activity relationship. So the relationship between the activity of a chemical molecule with respect to its structure. And these are called molecular descriptors, these values assigned to the molecules that they use to build these QSAR models. And topological indices are just a type of molecular descriptor. They have also based on other properties of the molecule, So we have the 2 graph theories so far. They developed independent of each other, although why do we call them graphs? I forgot to mention that. Back in the article back in 1878, where he enumerated all the saturated hydrocarbons, he referred to his drawings of his chemical diagrams as graphs, and it stuck. So actually, that's a chemical graph theory. Okay, so as I mentioned, in combinatorial graph theory, though, there was a large focus on max-min and combinatorial optimization sort of things. How far can you push the graph before you force this to happen? So these are quite different, because in chemical graph theory, they're computable. Like we just computed the Balaban index, where the domination number, for example, is not, if you have unless you have a small graph. But typically in graph theory, you're not looking at just small graphs. You want to let your graph be arbitrarily large, whereas in chemical graph theory, you're usually working with small molecules. So while I was looking at all of this is thinking about using graph theory to model protein structure, and we still need to keep looking, I think. So we'll keep looking. and go on to spectral graph theory. So spectral graph theory uses the Laplacian matrix in linear algebra. So the Laplacian matrix is the degree matrix minus the adjacency matrix. So you can see down the diagonal, these numbers are just the degrees of these. So there should be 1, 3, representing the vertex of degree 3. So your degree matrices, or your degrees down the diagonal, and then the minus ones, it's because you have minus the adjacency. And so in the adjacency matrix, you have a 1 in the ith row and jth column if vertex I is adjacent to vertex j, otherwise you have a 0. So if you're subtracting the adjacency matrix, you're getting this minus 1. Notice that if you add the rows, they add up to 0. And does that make sense why that would be true? Because you count how many edges, and now you're subtracting edge one at a time. Okay. So if you find the eigenvalues of the Laplacian, that's called the graph spectrum. And the number of zeros is the number of connected components. So you're really measuring the connectivity of the graph. And in fact, the second smallest eigenvalue is called the algebraic connectivity. So the algebraic connectivity of a graph is different than the combinatorial connectivity of a graph. So do you know what the definition of the combinatorial connectivity of a graph is? Anybody? It's either maximum or minimum something, isn't it? It's the minimum number of vertices that you can remove and disconnect the graph. So it's somewhat different. Okay, but we're still thinking about modeling protein structure. And so if we do a survey of the literature up to 2002, we can find examples, and in fact there is a nice survey paper that helps with the survey, Protein Structures Insights from Graph Theory, that surveyed different ways that graphs have been modeled, or proteins have been modeled with graphs. And usually it's the 3D, we're talking about the 3D structure and the proximity of these different parts of the protein. But if we look at the graphical measure up to 2002, the most popular was the algebraic graph theory.
So what's residue?
Yeah, we're going to get to that. I didn't know how to put what, but residue is the part of the amino acid, and a protein is made up of amino acids. Okay. Now, also in addition to protein structures, we find the other examples of where algebraic graph theory is used to model or describe or quantify a graphical representation of a biomolecule, like the secondary RNA structure here. So this group at NYU represents secondary RNA structure with a tree graph. And then similar to the saturated hydrocarbons, all possible configurations are given, and these are cataloged by their second smallest eigenvalue, or the Fiedler eigenvalue. Now, after 2002, we began seeing more and more what we refer to as network science. The small world phenomena paper came out in '98, and then the 6 degrees follow up by Watts in 2004, and we find definitions related to graph structure that are not in any of the 3 previously mentioned, combinatorial, chemical, or algebraic, because we're looking at a particular kind of graph. We're looking at a large graph, usually, that has a particular structure that emerges naturally. And that structure tends to have these dense pockets, and then the dense pockets are connected by a few edges. So what that gives you is that any pair of vertices, the distance is relatively small between them, even though the graph might be very large. It's like you can go from a hub to a hub and get to where you want to go. And so these terms, closeness, betweenness, centrality measures, and in particular the clustering coefficient, which is the denseness, and the average path length criteria for small world phenomena. We didn't see that in the first survey paper, but literature review post 2002, and you see a lot of networks appearing. So network analysis of protein structure, network networks, and even into 2013, we've got small world networks for studying protein structures. So what we have so far, we've got chemical graph theory. It's pros, it's well developed, it's computable, it's cons. Well, it's really, those values tell us, mainly things about a small graph or a small molecule. So computational chemistry and drug design is a big thing, because you're designing a small molecule. In combinatorial graph theory, it's well developed, but these measures are intractable. Spectral graph theory, also well developed, but it seems to have, it's useful, but still somewhat limited for all of biology. Protein structure in particular is what we're trying to focus on. Then inter-network science is now well developed, but it seems to be best for these large networks. And so it doesn't necessarily capture on the small scale. So since all of these, none of these alone are exactly what we need for our protein structure, you see a lot of specific applications of graph theory where terms have been defined or processes for that specific application, and it works very well. But that's also its con. It's for a specific application and not in general. So now to answer your question about what is a residue, so we can say, well, let's look at this more closely in modeling a protein structure with graphs. Let's have a quick overview of protein structure so we can then see how we might overcome some obstacles. So we'll talk about what proteins do, what are the levels of protein structure, how does the body make a protein, and the protein data bank. So proteins, if you think of the cell as like a city, a bustling city, and you've got trucks hauling cargo, and you've got people cleaning up the streets, and you've got people building buildings, and you've got people moving things inside and outside of the city, i.e. inside and outside the cell. Those are all proteins that are functioning. It's just like so when you're alive and you're functioning, your proteins are doing it all. That's what they do. There is an assumption that the function is highly related to its structure, although it's thing like anything in biology or the real world, it's not. It's the difference between probable and possible again. How are they made? The DNA sequence contains these segments we call genes, right? And then by a copy of the gene that encodes a protein is made called transcription, and that copy is our RNA. It looks just like the top string here, except where there's a T, there's now a U. Since this is a copy, it can now leave the nucleus and go to where there's the protein-making machinery. And every group of 3 here encodes for a specific protein. So as it reads this off, this RNA sequence encodes, say, this amino acid sequence, because each of these represent a different amino acid. So the structures are primary, secondary, and tertiary are the 3 structures of a protein, starting with the primary. That amino acid sequence that we just said that was created by the RNA sequence, that's the primary. So this is an example, say, of a primary sequence of a protein. It's just the list of the consecutive amino acids. Now, if you think of the string of amino acids, You can look at each one, and this is what each one looks like. What distinguishes one from the other is the R, and that's the residue. And so we'll look at that a little more closely, like right now. So this is what was in the circle. And notice that this is nitrogen, carbon, carbon, and they all have this structure. And when how they link up, not a biologist, so I'm going to say they link up. And it goes NCC, and then NCC, NCC, NCC. You get this chain. Eventually the chain stops, so on one side it's an N, and on the other side it's a C. And you have your N terminal and your C terminal. So this is just making the link, and this actually says what kind of amino acid it is, what's hanging off of it. So that's the primary structure. Then you get this long amino acid sequence, and so I circled this so we could say, well, suppose it starts here. Then it forms different structures, like this is called a beta strand, this is called an alpha helix. They don't really look like that, but this is our cartoon representation, right? So we can spot them. So the amino acids have these R's dangling off, and they're attracted, and they start to twist or bend or things. So one of the structures that it forms is the beta strand, and one of the structures it forms is an alpha helix, and those are the secondary structures. And then the whole thing in the three-dimensional molecule is the tertiary structure. So you have this underlying sequence, and then giving us this molecule. So you can play around in the protein data bank and have all kinds of fun, because they have all kinds of ways you can look at one. So on the left, it can be the same one. You're just visualizing here the ball and stick, of the molecule. Or over here, it's colored so that every single amino acid gets a different color, and you can see the amino acids that way. And it's a big problem. It's a fascinating problem that people are still working on. It's a protein folding problem, because if you know the gene, then you know the amino acid sequence it encodes. But you may not know its 3D structure. And so if you don't know its 3D structure, you're not sure what it does, because of that correlation between structure and function. So in the lab, biologists can crystallize the protein long enough to get a snapshot of its structure and map out the 3D coordinates of every single atom in that structure. And with that information, they put it in the protein data bank, and that's where we were looking at some proteins. But not all proteins are solved yet. We still don't know the protein structure, and so that's the protein folding problem. I believe so, because I think then it would mess up its function if but it could be in a different. Yeah, I should just defer to some biologists say that.
Yeah, generally speaking, yes, and evolutionarily it's been selected so that all of the many different possible combinations, the ones that have survived are the ones that adopt a particular structure. But yeah, the prions, there's one structure and then to another structure, which is infectious because it causes another one to have normal structure. But generally speaking, yes.
And that's important for long-term memory constitution. So it's not just for disease. Yeah. And also disordered proteins, there's a section which is not really folded. So, and often that interacts with a whole bunch of other proteins. So it's like, then they sort of hang off because
They're sort of the criminals in the city. No, they're disorderly.
Like in signaling, the molecular modes
Mm-hmm.
Ion channel middle is closed. So the crystallographic structure is pretty fixed, but there's no functioning of it. It's not necessarily the case. And there's all sorts of close translational applications like phosphorylation and glycosylation.
Yeah, it's complicated, but it's really interesting. All right, so this is the, I guess you call chemical diagrams of the 20 amino acids, and they're grouped into groups like, what do we got? Charged, negatively charged, etc. But one thing that we can notice here is this is the backbone. This is the part that we saw before that all of them have, that's identical on the top. So what these drawings have, I pointed to this is the nitrogen carbon that we mentioned before that forms the chain of the amino acid, and then the R that makes them all different. You can think of it as starting here, and you see the bottoms are what hangs off is different for each one. So since the top is the same, maybe it's just to advance, I chopped off the top. And then you can just see these and think of them as graphs. The corner points represent the carbon atoms. So if we look at the residue, these are the residues of each of the 20 amino acids, we can create a small graph for the residues. And given that these are small, then it's easy to go ahead and use some combinatorial graph theory as well as chemical, because the maximum degree is 3, longest path is 4, domination number is 2. I mean, they're real easy to calculate those values. But we have an issue, because no matter how much you calculate all the different graphical and right, what's going to happen here? There's no difference between these two, the residue Q and the residue E. We need more information. We need to know how they're different. And so how they're different is we have two oxygen atoms here, and we have a nitrogen over here. So in this case, it's glutamic acid, and over here, it's glutamine. So we need to figure out a way to use graph theory, but also have this information in there if we're going to model these amino acids with graph theory or with graphs. So I think that's just what I said. So the next one. So let's think about the graphical invariants from graph theory. Like this is degree 3. There's a vertex V and it has degree 3. Because what we say is it has 3 edges incident to it usually. You could say, or it has 3 neighbors, or it's adjacent to 3 vertices. But if you go ahead and label the vertices with a 1, an understood 1, then you could alter the way you're saying the degree of a vertex by saying the degree of a vertex is the sum of the weights of its neighbors. And it's the same definition, and thinking of labeling them 1 is consistent with what we usually do. X with no exponent, there's an understood 1, right? No coefficient, an understood 1. So if there are no labels on the vertices, there's an understood 1. So that's perfectly fine. And so now we can change our definitions to almost all of the graphical invariants and reduce them to special cases when the weights are just 1. And so if the weights are not 1, this vertex now has degree 7. So going back to our issue before, that changes the degrees for those guys. Okay, so the domination number is 2 because you can dominate this graph with 2 vertices. The 2 black ones, we can say all the remaining vertices are adjacent to at least one of the black ones. And you can do it with fewer than 2. So 2 is the minimum number that you need to dominate the graph. But now if you find all dominating minimum cardinality dominating sets and then add the weights and take the minimum weight, that could be your weighted domination number. So that's what we did for one of our first projects. Is we have these two are based on vertex weights, and this one's based on vertex weights. This is just the number of cycles. You may have noticed on the residue, a few of them had a cycle structure, but most of them didn't. And we also had to include like some biochemical properties, hydrophobicity, and their charge. And see, this was the Q and the E before. One of them's negatively charged and one isn't, but there's a slight difference here and here because of those vertex weights. Now, why do we want to do that? Well, if we're looking at the primary sequence, which is a sequence of letters, that doesn't give you much information about how they compare to each other. But if you have Q and E, you can see that numerically, just like they are physically, they're close. They're not the same, but they have things that are in common, where some of these are quite different. So we can change this alphabet sequence to a numerical sequence where we take this string and replace L with that string, replace L with the same string. Notice that G is all zeros because G doesn't have a residue. Some people think of it as a spacer or something. So you can now do more computation with these numbers. And we, as a guide, we were trying to use the properties of these amino acids. So like the hydrophobic or F, Y, H, and W, and just doing a simple clustering with the ones that I had up there, you get this. Even without the hydrophobicity, it changes a little when you add more. But just trying to capture numerically the distinguishing properties of the 20 amino acids. And then there was this dream challenge. I'm not sure when it was. I don't remember either, but it was a while back. I think it was Dream 4. And DREAM stands for Discussions in Reverse Engineering Assessment Methods. So like the CASP and the other different challenges they put out there to the computational community, they have the answer. Will your algorithm, how close will your algorithm get to the answer? And the DREAM challenge is organized by Gustavo Stolovitsky and Andre Califano at IBM and Columbia, respectively. The year that we participated, Robert Prill and Julio Rodriguez, I don't know if they're still involved, but certainly Gustavo and Andre are still running the DREAM challenge, which is one ongoing right now. But the one that we participated in was this. Here's the primary sequence of a protein, and this protein binds to this protein. And this is the PDZ domain for the other protein, the target protein. So this sequence recognizes and has high affinity for this sequence. So they bind, and then, because often proteins will come together and bind together to form a protein complex to do a certain job, and then when that's job, they run off and work with somebody else. So they have this interesting or amazing how they can, what all happens inside the cell. So this sequence has affinity for this one, this has affinity for this one, and so forth. And all of this is given to you. And here's the challenge. If you're given this sequence, what does it have high affinity for? What will it most likely bind to? What sequence over here will this want to make, bind to that one? And see, I put this in bold so we could see that ELGF, what they did is they did point mutations here and there. And then in the lab, they have the answer to this. And what the challenge is you return a matrix that has the probability of which of the 20, probability measures for each position. that will make this be the sequence that it binds to. And so they also provide other, sources of data for you and your algorithm. So what we did was Jeff, and we worked on this together, and he wrote this artificial neural network. These are the ones that we had on the preceding slide that we can convert an amino acid sequence to a numerical sequence. And so we gave it the proteins and the target proteins, and told it, yes, this is, using the data, that this is attracted to that, or no, it's not, till the artificial neural network could recognize the patterns. And then again, the input was just from this graph-theoretic models using vertex weights. And it came in second. So it was our first shot, the first time to try these vertex weights. And so we were tickled about that. And without the vertex weights, it wouldn't have worked. And we tried to I don't know what you did to try. If there weren't any vertex weights, what did you put in?
I just put in 3D.
Oh, okay. Okay, but that's the primary structure. Still looking for, say, a model for the tertiary structure. And this one that we're looking at here, I believe, is the NBD1, the nucleotide binding domain 1 of the CFTR, the cystic fibrosis transmembrane conductance regulator. Now, the goal is to model CFTR with a graph. But here's the issue. CFTR has 1,482 amino acids. It's a huge molecule, right? And that's 1,482 amino acids. And each amino acid has, 10, 12 atoms. So, you're looking at close to 15,000 vertices if you're going to let each vertex represent an atom. So that might not be too good. Or, close to 1,500 if each vertex represents an amino acid. Now, CFTR is the protein associated with cystic fibrosis. There is a single amino acid out of the 1,482 at position 508 that's deleted. So just this one is deleted. And because of that missing amino acid, this protein doesn't fold correctly. So it's recognized by the cell's quality control mechanisms that, this isn't doing what it's supposed to do. We're going to chunk it and try again. But because of that one amino acid, it just never is produced. So people with cystic fibrosis don't have this particular protein in their body. If you don't have this protein in your body, that means that wherever you have mucus, like in your lungs or in your intestines, wherever you have mucus, it doesn't have the right viscosity, because this protein's job is to monitor the salt in the cells where the mucus is and make sure that the mucus has the right viscosity. And it has too much salt because this protein is missing, so the mucus is just sticky, and it does the opposite of what it's supposed to do. Instead of being, helping things clean, like when you cough, you clean your lungs, it just sits there and gums up the works. One amino acid out of 1,482. Now, how are you going to model that? See, if you let every vertex be an amino acid, and you've got almost 1,500 of them, and you say, okay, now I'm going to delete it, it doesn't really change the graph that much. Right? So that's the issue, because it's the dominant. Even if you weight the vertices, it's just one. It's not going to change these global measures that much. Okay. Here was the idea. Start at the top and say, all right, let's take the graph, or the molecule, this big protein molecule, and break it into subregions. So this is not the entire CFTR. This is just one of the 5 domains that CFTR has, this NBD one. But let's break it into smaller regions. And then each region is, think of it as a smaller 3D molecule where it's small enough now that we can think of each vertex as an amino acid. So these are now amino acids. And the amino acids that are close enough to each other in a 3D structure will go ahead and connect with an edge. Those that aren't, we don't. So we're capturing the 3D structure of the interaction of these amino acids and just in this little region here. But then, of course, each amino acid is a graph, as we've seen, and we just used in the Dream Challenge. So really, you have a graph inside a graph inside a graph. And then, if you want to think about a single point mutation, which is what we want to do with cystic fibrosis, then, you know, well, we'll keep going. This is the idea, so we'll look at it specifically. So here was this case. We took NBD1, broke it into 8, subregions. So they each have their own primary sequence. And there's an example of what the graph would look like. There is the one that causes so much trouble when it's deleted, F at position 508. So now we can do like a virtual mutation at this level. If we take out 508, we could just take the resulting graph. Now, I know there are lots of biological problems with that. You're going to change all of that. But just, as a starting point, if we were to just remove 508 and its edges, then we also, these are amino acids, so we can weight the vertices and calculate things based on those vertex weights. And so the global measures will change when you pull out 508 because you've lost one of your weighted vertices. So these were the mutations that we were looking at. And so just have them black and red. This means that, which one is it? That's this one. The amino acid G at position 542 was replaced with an A. And so that's just a repeat that there is the F in this particular substructure, and now we're going to do a virtual mutation. And wanted to put this back up again because now that we have more background, see, if we take out F or we take out whichever one, if that's gone, you change your graph, and so you change this global measure, which results in a change of simply one vertex measure here. But that's appropriate. Here you change the whole thing. Here you change some of this, and now you're just changing one, a weight on one of these. So it's kind of trying to take that information and filter it up to the whole protein. So that's what we did, just using those same amino acid descriptors, those same weights that we'd used before, maybe put a few others in. And then that gives you for each, you do your mutation and you recalculate it again, do your mutation, recalculate it again based on those vertex weights. And so it's a good start because these are fairly mild, is the belief. Whereas there's 508 and G551D are both very, cause severe consequences. So the conclusion, there are lots of examples in the literature where graphs are used to model biomolecular structure. The vertices can either be atoms, amino acids, secondary structures, but we have the problem that, we have the problem of different scales. So you've got, the amino acid to the protein molecule, but you can even keep going. Then you got a protein that's part of a protein-protein interaction network. And so how can a change at one scale affect the system at the other scale? And vertex weight seems to hold a promise as a possible way of doing that. So what kind of graph theory should we use? Well, I think all of them, because we used a mix of chemical and combinatorial and then network science and spectral graph theories in there, so you can't have any favorites. It's just all graphs, right? And bring it all to the table. And at this level, it might be chemical. At this level, it might be combinatorial. At this level, it might be best to use network science, but you should keep pushing that up. And what all of this put together in the age of data science, all of this put together, it's graph analytics. That's it, I believe. Yeah.